So let's go ahead and take a look now at the actual synapse itself and the neurotransmitters that are going to function at those synapses. So the synapse is, like I said before, a specialized gap junction that's going to provide a means for regulating the transmission of the communication signals, where we're going to have a space or a synaptic cleft that is going to be needed to cross in order to get the message signaled from one neuron to the next. Primarily, these are going to be chemical, and to a lesser extent, we get the electrical. And like we talked about before, the more we use them, the stronger they'll become, and the less we use them, the weaker they'll become. And if we get excessive weakening, it will actually lead to that apoptosis because the secretory cell the neuron wants to secrete. And if we're not giving it a chance to secrete, then it's going to undergo its apoptotic cycle. The transmission itself is the way in which we can take the electrochemical charged action potential and take it from one neuron to the other neuron. And so when we look at this, we're going to look at it based off of the chemical signal, and then we'll talk about in more detail when we get together during our online sessions, the electrical plasticity issue here. So all synapses start chemical. The more we use them, the more desmotic those junctions become as we get more proteins adhering the membranes together. And eventually, over time, they're going to form simple electrical pathways, meaning they're going to act as a single cell. So we're going to walk through the way in which we go from the action potential now to the chemical transmission. So the way in which this happens is that we start with an action potential that's going to reach the terminals. When it reaches the terminals, it opens up calcium-gated channels, allowing calcium to influx into the neuron. This calcium influxin is going to cause vesicles holding the neurotransmitters to bind to the membrane itself. As it binds to the membrane, that membrane is then going to exocytotically release the neurotransmitters into the synaptic cleft, where via diffusion principles, we're going to diffuse across the synapse, bind onto the chemical receptor for that neurotransmitter, and then cause the postsynaptic event. Following the postsynaptic event, that receptor will release the neurotransmitter. That neurotransmitter that can diffuse back across the synapse, where it can be taken back in via reuptake at the presynaptic membrane. It can be digested within the synapse via enzymes. The digested neurotransmitter can either be taken in at the postsynaptic membrane or once again be reuptaked at the presynaptic membrane. Any neurotransmitter that gets taken back in by the presynaptic membrane will get recycled into a vesicle. If it's been broken down via digestion, it will get reassembled at the terminals and then vesicled into the neurotransmitter vesicle. So let's take a look at how this goes about functioning.
At a chemical synapse, a neurotransmitter molecule, such as acetylcholine, communicates a signal from one neuron, the presynaptic cell, to another, the postsynaptic cell. Neurotransmitter molecules are stored in vesicles in the
So that's how the synapse works. But what's the function of the synapse? So the synapse is going to act as a relay checkpoint. It's going to make sure that we get the correct signal going through the pathway. It's going to establish a delay in the pathway so that the end result of the activation is timed correctly to when a response is necessary. In this pathway, we can have amplification or we can have muting of the signal based on the way in which the impulse is being processed. In amplification, we get a larger signal coming out through the pathway than what went into the pathway, meaning that we're getting a longer action potential train coming about. In muting, what ends up happening is that we're not actually stopping the action potential train, but we're diminishing the total numbers of impulses being passed along. In this, we have the impulse processing cells, where what we're doing is the convergent and divergent neurons within the neural networks are pooling all of the signals together, or we have neurons within the pathway that are regulating receptors based off of the need to be activated. And what this impulse processing does is it causes that amplification, or it causes that muting of the signal that comes about. In part of this processing process, what ends up happening is we get distinct postsynaptic activations or postsynaptic actions that take place based off of what type of signal and what type of impulse processing is taking place. In this, we can have what's referred to as a facilitation on a neuron-to-neuron pathway, in which we have two options. Either we are inhibitory postsynaptic potential, IPSP, or we are excitatory postsynaptic potential, EPSP. If we have multiple neurons coming together, we have distinct types of integration, or what's referred to as summation, that can take place. If it happens based off of a timing of signal, we get what's referred to as temporal summation. If we get it based off of areas of the neurons being interactive with each other, it's what's referred to as spatial summation. Within the pattern of neuron interactions, we can have convergence and divergence of the actions. Convergence, we are taking multiple neurons and funneling them onto a single loci or nuclei. Divergence, we're taking that single nuclei of neuron and we are splaying it out to multiple endpoints within the network. Based off of impulse processing, there are two options that can come about, and we'll get more detail when we talk about sensation processing. And this is desensitization, and that is the loss of the sense over time that can either be temporary or permanent. And then attenuation, which is where the stimulus is still being processed, but the cognitive response to it has stopped. If we look at the way in which patterns come about, we have the two types of patternings that are going to lead to the postsynaptic excitatory potential, where we're going to increase signal propagation, or postsynaptic inhibitory potentials, we're going to diminish signal propagation. The first is temporal. In temporal, we have trains of excitation and trains of inhibition, in which if we have the train of excitation summating and generating more excitation than inhibition over a set amount of time, we're able to get to threshold and propagate the signal. So temporal is all about timing, as in when does the excitation versus the inhibition strike the neuron. If the neuron is in refractory when the excitation comes about, it will not be passed. If we have inhibition of the neuron, we need to have additional excitations in order to get above threshold. The other type of summations are the spatial summations. In this case here, we have two types of spatial summations. We have soma summations, which is the top one here, in which we get multiple neurons converging and synapsing all at the same time, attempting to pass their signal along. Where if more of the neuron soma is able to reach threshold, then the action potential will be sent. Whereas if less of the soma is able to get to threshold, we will not get that action potential being sent. The other type of spatial summation is based off of the synapse, in which we have postsynaptic and presynaptic inhibitory signals. On the presynaptic inhibition, the spatial summation puts the axon terminal of the presynaptic membrane into refractory so that the action potential coming down is unable to pass along. When we're looking at spatial summation, it's about, can the membrane get to threshold based off of the signal arriving at the same time? Whereas with temporal, it's, are we in refractory when the next signal comes into play? And so regardless of the type of summation, what we're talking about is, can the membrane get to threshold, or is it stuck either in a hyperpolarization or in a refractory within the polarization? The way in which we're able to get the summation is off of the neurotransmitters, which we'll talk about in a second here.